students, and today we are going to discuss about soil microbiology. That means study of microorganisms which are present in soil is called as soil microbiology. So, soil microbiology means study of organisms like include bacteria, fungi, algae, virus, actinomycetes, protozoa and etc. Okay. So, how these organisms play a role in soil, that is the thing we are going to discuss today. So, it is a branch of science deals with study of soil microorganism and their activities in soil. So, their functions and how they affect soil properties from a very small fraction of soil mass. Okay. In the upper layer of soil, that is top soil up to 10 to 30 centimeter depth, that is called as horizon A, microbial population is very high, which decreases with depth of soil. So, you may know that in upper surface it is directly related with the environment. So, it contains most of the favorable conditions for the growth of soil, whereas in goes depth there will be a change in the survival conditions. That is why it leads to the less number of microbial population in depth of soil. Living organisms, both plants and animals, constitute an important component of soil. So, the pioneering investigations of number of early microbiologists showed for the first time that the soil was not an inert static material, but a medium of pulsating with life. That is, the soil is highly fertile, which favors the growth of organisms. The soil is now believed to be a dynamic or rather a living system containing a dynamic population of organisms and microorganisms. Then cultivated soil has a relatively more population of microorganisms than the fallow land, and the soil is rich in organic matter which contain more population than sandy and eroded soil. The importance of microorganisms in soil, why it is needed, means it affects the structure and fertility of different soils. They contribute to nutrient availability in soil, that is organic matter decomposition, humus formation, nitrogen fixation and seed germination. And also they manage soil stability by different biochemical process, and also they involved in the degradation of pesticides and chemicals in soil, and they contribute the growth and success of plants and overall ecosystem of soil environment. And moreover, microbes in the soil are important to us in maintaining soil fertility and productivity. The nutrient cycling of elements in biosphere also done by the microorganisms. They are the rich sources of industrial products such as enzymes, antibiotics, vitamins, hormones, organic acids, etc. And at the same time, certain soil microbes are the causal agents of human and plant diseases. So, what are the soil organisms? So, maximum soil contains more number of microbial and macrobial flora and fauna, and soil flora and fauna, and which includes microflora, that is bacteria, fungi, actinomycetes, algae, and macroflora includes root of higher plants. And soil fauna contains protozoa, nematodes coming under the category of microfauna, earthworms, moles and ants are coming under the category of macrofauna. The soil inhabit the several diverse group of microorganisms, but most important among them are bacteria, actinomycetes, fungi, algae and protozoa. And bacteria is ubiquitous in nature, that means where everywhere it is present, which inhabits in soil also. Bacteria are the most abundant and predominant organism in soil, and these are primitive organism which are classified into prokaryote, and they are microscopic and unicellular microorganisms without chlorophylls. So, morphologically soil bacteria are divided into three groups: cocci, rod shaped, and spirilla. Bacilli are most numerous organism, followed by cocci and spirilla in soil. So, the most common method used for the isolation of soil bacteria is dilute plate count method, which allows the enumeration of only viable counts in soil. The size of soil bacteria varies from 0.5 to 1 micron in diameter and 1 to 10 microns in length. They are motile with locomotive organs of flagella. The population is the highest population in microbes, that means 1 million, more than 1 million to several hundred millions per gram of soil, depending upon the physical, chemical and biological conditions of the soil. So, Vinogradsky, on the basis of ecological characteristics, classified soil microorganisms in two types, generally autochthonous and zymogenous. So, autochthonous is nothing but indigenous organisms which are native to that environment. Bacterial population is uniform and constant in soil, since the nutrition is derived from the native soil organic matter. Zymogenous bacterial population in soil is very low, as they require an external source of energy, example Pseudomonas and Bacillus. So, the population of zymogenous bacteria increases gradually when a specific substrate is added to the soil. So, to this category belonging to cellulose decomposers, nitrogen utilizing bacteria and ammonifiers. So, bacteria are also classified on the basis of physiological activity or mode of nutrition, especially the manner in which they obtain their carbon, nitrogen, energy and their nutrition requirements. So, they are broadly divided into two groups, one is autotrophs and another one is heterotrophs. So, autotrophic bacteria are capable of synthesizing their food from simple inorganic nutrients which are available in soil. So, heterotrophic bacteria depend on preformed food for nutrition, that means they depend the autotrophic organism. So, all autotrophic bacteria utilize carbon dioxide, which is absorbed from the atmosphere as a carbon source, and derive the energy from sunlight, okay, and involved in the photosynthesis and produce the organic compounds. So, the example for photosynthetic organisms are Chromatium, Chlorobium and Rhabdopseudomonas, or they obtain the nutrients from oxidation of simple inorganic substances which are present in the soil. They are called as chemoautotrophs, example Nitrobacter, Nitrosomonas and Thiobacillus. So, majority of soil bacteria are heterotrophic in nature and derive their carbon and energy from complex organic substances or organic matters and decaying roots and plant residues. So, they obtain their nitrogen from nitrates and ammonia compounds which are present in soil, and other nutrients from soil or from the decomposed organic matter. So, certain bacteria also require amino acids, B vitamins and other growth promoting substances also for their growth and activities. So, the bacteria bring about a number of changes and biochemical transformations in soil, and thereby directly or indirectly which helps in nutrition of higher plants growing in the soil. So, the important transformation process in which soil bacteria plays a vital role are decomposition or ammonification or nitrification or denitrification and biological fixation of atmospheric nitrogen, oxidation and reduction of sulfur and iron compounds. So, by this way that all nutrients are transformed from soil to organisms. So, some of the examples are given in this table. Okay. These are the organisms which are involved in the decomposing of cellulose compounds. And next to bacteria, actinomyces takes place the major part in soil. So, they are the organisms with characteristics common to both bacteria and fungi, but yet possessing distinctive features to delimit them into distinct category, and nowadays it is confirmed with bacteria. So, that is why it is called as actinobacteria. And in the strict taxonomic sense, actinomycetes are clubbed with bacteria. The same class of schizomycetes are confined to the order of actinomycetales. So, they are unicellular like bacteria, but produce a mycelium which is non-septate and more slender. Like true bacteria, they do not have distinct cell wall, and their cell wall is without chitin and cellulose in the cell wall of fungi. So, on culture media, unlike slimy distinct colonies of true bacteria which grow quickly, actinomycetes colonies grow slowly, so powdery consistency and stick firmly to the agar surface. So, when compared to bacteria, the growth of actinomycetes takes slow and more time for their growth. So, they produce hyphae and conidia or sporangia like fungi. Certain actinomycetes whose hyphae undergo segmentation resemble bacteria both morphologically and physiologically. Actinomycetes are numerous and widely distributed in soil and are next to bacteria in abundance. They are widely distributed in the soil complex, compost and etc. And it estimates that 10 to the power of 4 to 10 to the power of 8 per gram of soil, and they are sensitive to acidic pH and waterlogged soil conditions. So, the population of actinomycetes increases with the depth of soil even up to horizon C of soil prolifer. They are heterotrophic, aerobic and mesophilic, which are growing under the temperature of 25 to 30 degrees Celsius, and some species are commonly present in compost and manures, which are coming under the category of thermophilic, which grows at the temperature of 55 to 65 degrees Celsius. The example for this thermophilic actinomycetes is thermoactinomycetes and streptomyces. Actinomyces belonging to the order of actinomycetales, which are grouped under 4 families: Mycobacteriaceae, Actinobacteriaceae, Streptomycetaceae and Actinoplanaceae. Actinomycetes genera, which are agriculturally and industrially important, are present in only 2 families of actinomycetes and Streptomycetaceae. In order of abundance in soil, the common genera of actinomycetes are Streptomyces, Nocardia, Micromonospora, Actinomycetes, Actinoplanes, and Micromonospora and Streptosporangium are also generally encountered in soil. So, what is the role and how it plays a major role in soil means, with these actinomycetes are involved in the degradation or decomposition of organic substances, and organic residues are first attacked by bacteria and fungi and later by actinomycetes. They decompose or degrade the more resistant and indecomposable organic matters and produce a number of dark black to brown pigments which are contributed to the dark color of soil humus. So, they are also responsible for subsequent further decomposition of humus or a resistant material in soil. They are also responsible for earthy or musty odors envelop freshly ploughed soils during the rainy season. And many genera, species and strains of Streptomyces, actinomycetes produce a number of antibiotics. So, one of the species of actinomyces, Streptomyces scabies, causes the disease in potato. Name of the disease is called as potato scab diseases. And next organism is fungi. So, fungi in soil are present in mycelial beds, rhizomorphs or as different spores. So, their number varies from few thousand to few milligram per gram of soil. They possess filamentous mycelium which is composed of individual hyphae. The fungal hyphae may be aseptate or septate. So, example for aseptate fungi, Mastigomycotina and Zygomycotina, and septate hyphae example is Ascomycotina, Basidiomycotina and Ditromycotina. C.K. Jackson observed most commonly encountered genera in fungi in soil are Alternaria, Mucor, Aspergillus, Penicillium verticillum, Cladosporium, Trichoderma, Cephalosporium botrydis, Rhizobus, Chaetomium, Glycolidium, Fusarium, Monilia, Pythium and etc. So, most of these fungal genera belonging to the subdivision of Ditromycotina are fungi imperfect which lacks sexual mode of reproduction. So, as these soil fungi are aerobic and heterotrophic, they require abundant supply of oxygen and organic matter in soil. So, they are dominant in acid soil because acidic environment is not conducive or suitable for the existence of either bacteria or actinomycetes. So, fungi takes place more growth in acidic soil. So, the optimum pH range for fungi lies between 4.5 to 6.5. They are also present in nutrient alkaline soil and some can even tolerate beyond the pH range of 9. So, what is the role of fungi means? How do the fungi play a role in means? Just they are involved in the decomposition matter or plant nutrition decomposition, lignin degradation, and they serve as a food bacteria. Apart from this, some fungi belonging to the subdivision of Zygomyconon and Ditromyconon are predaceous in nature and attack on protozoa and nematode in soil and thus maintain the biological equilibrium in soil. So, they also plays a role in important role in soil aggregation and in the formation of humus. Soil aggregation means just they accumulate the nutrients in particular area for the development and growth of plants. Some of soil fungi are parasite and causes number of plant diseases such as wilt disease, root rot disease, damping off and seedling blights. These type of diseases are caused by some of fungi, Pythium, Piphilophora, Fusarium, Verticillum etc. And number of soil fungi forms mycorrhizal association with the root of higher plants, that makes symbiotic association of fungi with roots of higher plants and helps in the mobilization of soil phosphorus and nitrogen. The example for symbiotic fungi are Glomus, Gigaspora, Aglospora and Amanita, Poetes, Endoloma, Lactarius. So, these are all coming under the category of mycorrhizal category fungi. And then next organism is algae, which are present in the waterlogged conditions. Most of the soil are where moisture and sunlight are available, there algae are present. Their number in soil usually ranges from 100 to 10,000 per gram of soil. So, they are photoautotrophic aerobic organisms and obtain carbon dioxide from atmosphere and energy from sunlight and synthesize their food. So, they are unicellular, filamentous or colonial structures. Soil algae are divided into 4 main classes: Cyanophyta, Chlorophyta, Xanthophyta and Basiloryophyta. So, out of these 4 classes, phyla, blue-green algae and grass-green algae are more abundant in soil. Green algae prefer acid soil, while blue-green algae are commonly found in neutral and alkaline soils. So, the most common genera of green algae are Chlorella, Chlamydomonas, Chlorococcum, Protostaphion and that of diatoms are Navicula, Pinularia, Sintra and Frangillaria. So, blue-green algae are unicellular phototrophic prokaryotes which contain the pigment phycocyanin in addition to chlorophyll. So, they do not possess flagella and do not reproduce sexually. They are common in neutral to alkaline soils. The dominant genera of blue-green algae are Crococcus, Formedium, Anabina, Aphanocapra and Oscillataria. Some blue-green algae possesses specialized cells known as heterocyst, which is the site of nitrogen fixation. It fixes the nitrogen in paddle or paddy water and waterlogged paddy fields. So, almost 20 to 30 kg of nitrogen per hectare in one season is fixed by these blue-green algae. There are certain blue-green algae which possess the character of symbiotic nitrogen fixation in association with other organisms like fungi, mosses, liverworts and aquatic ferns. Azalo. Example is Anabaena azola association, which fixes the nitrogen symbiotically in rice fields. So, how do they play a role means it maintains the soil fertility, especially in trophical region. They involved in the addition of organic matter to soil when die and increases the amount of organic carbon. Most of the soil algae are acting as a cementing agent in binding soil particles and thereby reduce the prevent soil erosion. And mucilage is the component secreted by blue-green algae is hygroscopic in nature and thus helps in increasing water retention capacity of soil for long period. Soil algae through the process of photosynthesis which liberate large quantity of oxygen in soil environment and facilitate the aeration in submerged soil and oxygenate the soil environment. They also involve in the helping of checking the loss of nitrates through leaching and drainage, especially in uncropped soils. And they involved in the weathering of rocks and building up of soil structure. And the last organism is protozoa, which is present very less quantity in soil, almost 10,000 to 1 lakh per gram of soil. They are unicellular, eukaryotic, colorless and animal-like organisms. And they are larger than bacteria and size varying from few microns to few centimeters. They can withstand the adverse soil conditions as they are characterized by cyst stage in the life cycle. So, except few genera which reproduce sexually by fusion of cells and the rest of them reproduces asexually by fission and binary fission. Most of the soil protozoa are motile by flagella or cilia or pseudopodia as a locomotor organs. So, depending on the type of appendages provided in locomotion, protozoa are rhizopoda, mastigophora, siligophora and sporophora. Then rhizopoda consist protozoa without appendages usually have naked protoplasm without cell wall, and pseudopoda used as the temporary locomotory organ are present in sometimes. So, the important genera coming under the category of rhizopoda are amoeba, Biomixa, Euglifia. And mastigophora belongs to the flagellated protozoa which are predominant in soil. Important genera of mastigophora are Allanton, Bodo, Cercopoda, Cercomonas, Entosiphon, Spigromonas, Spongomonas and Tetrametus. So, many members are saprophytic and some possess chlorophyll and are autotrophic in nature. In this respect, they resemble unicellular algae and hence are known as phytoflagellates. The soil protozoa belonging to the class Ciliata and Silicophora are characterized by the presence of cilia. Cilia is nothing but it is a short hair-like appendages which around their present around their body, which helps in locomotion. So, the important soil inhabitants of this class are Colpidium, Calpoda, Galantophorus, Gastrotila, Halteria, Euroleptus, Verticilla and Plerotricha. So, protozoa are abundant in the upper layer of soil organic manures and they involve the soil moisture, aeration, temperature and pH. These are the factors which affects the soil protozoa. And what is the function of role of protozoa means most of the protozoans derive their nutrition by feeding or ingesting soil bacteria belonging to the genera Enterobacter, Agrobacterium, Bacillus, Escherichia, Micrococcus and Pseudomonas, and they play an important role in maintaining microbial or bacterial equilibrium in soil, and some protozoa have been recently used as biological control agents against phytopathogens, and some species of bacterial genera, Enterobacter, Aerobacter, are commonly used as the food base for the isolation and enumeration of soil protozoans. Several soil protozoa cause diseases in human beings which are carried through water and other vectors, for example amoebic dysentery, which are caused by Endamoeba histolytica. So likewise, most of all microorganisms plays an important role in the nutrient cycling, decomposition, nitrogen fixation, carbon dioxide fixation, so sulfur fixation. So likewise, the microbes play a major role in soil environment. Thank you.